co-chairman and the rapporteur, and my dear students and the other dignitaries present in the audience. Usually speaking after the lunch is very challenging because everybody feels sleepy, and so do the speaker. But I have come to you with a presentation, and this presentation I have made specifically looking at the students, and I have kept my tag out. I am not a director. I am one of you. I'm still a student, and I'll be talking about some new hypothesis of the new way that we are trying to look at the spermatozoa. And I have kept my presentation very simple, and sometimes I have oversimplified this for the sake of understanding and giving it to your brain. And of course, but whatever I'll be hypothesizing, so I'll be speaking with the data that has been generated at my laboratory. So this is time that we look at this spermatozoa beyond their motility. We have come a long way, and this theory of simply counting the spermatozoa or looking at their motility is probably not working anymore. Although we are currently predisposed or remain obsessed to look at the, and why not? Spermatozoa is a very, very fantastic and very interesting cell because it moves. And if you look at the mitochondrial architecture of the spermatozoa, this is so unusual that this particular cell, it generates energy for itself to proceed forward, and this is probably required. And if you look at the journey of the spermatozoa from its site of deposition to the site of fertilization, this is again, I mean, there is no race that is tougher than this. So let us start with this. So I thought that I'll be presenting under these headings. I'll be talking about the current perception of the semen quality and what all the sperms they carry, and there is smart sperm versus the non-smart sperm. Suddenly, with the current understanding, we are understanding the spermatozoa, they are of two kinds, just like us. Some spermatozoa are very smart and some are not so smart. And then our evidences on the genomics and the proteomics and the expectation from a good sperm, what all we expect from a good sperm. And at the end of the day, I'll be coming to some take-home message of the conclusion. This is a kind of well-known fact, and the more and more pressure we have kept on the animal, so the animals got compromised with respect to their fitness traits. So nothing comes, there is no free lunch in this particular world, right? So when you put a pressure on one hand, the other hand gets a kind of compromise. For example, when you say fitness traits, they are disease resistance, they are fertility, etc. And if you look at the history of the milk-producing animals, be it a buffalo or Holstein Friesian or whatever, you find that over the last 50 years, they have been compromised with respect to their fertility. In case of the female, we say they are kind of the estrus behavior, and in case of the males, we say their ejaculate rejection rate, etc., etc. My colleague Dr. Pradeep knows better about this. So what we can do about it? If we look at it, so these are the classical yardsticks to measure the fertility, their fertility. What we look at the spermatozoa in terms of their morphology, their motility, and their count. These three things. You go to any semen lab, you find these three criteria. They are used again and again. So the evaluation of the male fertility, I say, the analysis of the semen, whenever we get a semen, we first look at its color, we look at its volume, we look at its pH, and then we put it in a spectrophotometer to know that how many of the sperms are there. But then the question comes that why do some of the prized males have no fertility, even when the classical semen parameters all have been used on them? I'll give you evidence. This is the evidence of a project that we run at CIRD over the last maybe 30 years, and this is the data of last 23 years. What we find? We find that out of the 127 pools that we measured, the fertility in some cases they were as high as 65%, whereas in some cases this was as low as 15-20%. And what I'm saying is, this is not a small or isolated piece of data. I have purposefully kept only those data, but the number of inseminations are at least 50. For example, this particular bull, here the number of insemination was 100 plus, and this bull, the highest one, here the number of insemination was more than 212. So we have some reason to believe this data. But the question comes that when these animals were used in the farmer's place or to the stakeholders, then we come to know that this particular bull is stupid only when sufficient damage has been done. So the hypothesis that I am going to present today is, can we pre-select? Can we pre-select the bulls? Or does anything exist like a good bull or the bad bull? Or what should we concentrate on? Whether we should concentrate on a bull or we should concentrate on ejaculate. Because on a given day, maybe coming Friday, a good bull will give you very bad semen. But then when you take it granted that this has come from a good bull and you freeze them, then there is a possibility that you end up in this kind of situation. What to do about this? There are some very simple arithmetic. You can take a calculator and count. What is this? This is every bull that gives you 2 ejaculates. And they give it twice in a week. And then 4 weeks, 12 months, and theoretically, a bull can produce semen for 10 years, 10 long years. So this ends up in so and so number of ejaculates. For buffalo, if I consider the average ejaculate volume is 2.5 ml, so then gives you about 4800 ml, about 5 liters of this semen. And then if I assume that they have about 1,000 million of the spermatozoa per ml, then gives you so and so 4.8 trillion of the spermatozoa. I do not consider all of them are live, but to assume that at least 70% of them are live, it's a good assumption. So then gives you so and so trillion live sperm. And as per the current practice, we freeze, we pack about 20 million of the spermatozoa per dose of the frozen semen. This is what is the central point which we are challenging. We are saying that why to keep 20 million spermatozoa, while probably a lesser number can do well. I'll give you evidence for that. So then we do that with 20 million with the assumption again that after the freezing, some will die. So at least 8 to 10 ml of the live spermatozoa will be still available for resulting into an acceptable rate of fertility. But then with the 6 semen era, we are finding that even a 2 million spermatozoa is enough, right? Although it requires some manipulation in terms of where we deposit that particular spermatozoa, their method of insemination, etc., etc. But 2 million spermatozoa per dose is possible. So we are arguing here is that every sperm is important. Let us not waste this spermatozoa. What is the test for a good sperm? The test for a good sperm so far, what we know is that the sperm membrane should be intact. We have many tests available for that, like hyposmotic soiling test, blah blah blah. And then it should be able to see a freezing shock, and post-thaw motility should be good, acrosome should be good. Please give me a reminder at 12 minutes. No? And then the sperm fertilizing ability and the karyotype. Yes, for every bull we do a karyotyping to prove that this particular bull is normal. So what is the missing link? When we do all of this, still at the end of the day, why we find such a fertility divide that some bulls are 70% and some bulls are 30%? So there is a paper which was published, I think, sometime in early 2012 or 2013, that counting spermatozoa does not add anymore, and this is time for a new equation. And this particular picture, this is called the ICSI, or the intracytoplasmic sperm injection. This is a very much accepted technique now as an assisted reproduction technique. So this again says that a single sperm is enough. Okay, so the issue is how to select this particular spermatozoa which we feel as most eligible. A lot of clue can be generated if we understand this particular picture. When the fertilization event happens, the spermatozoa, they know that we have to do these functions. I say here that the sperm remains pre-programmed to govern all of these events. What are these events? It's that the spermatozoa, it has to approach the madam egg, and then it is said the spermatozoa, this has to fuse with the egg cell, then it has to penetrate through the zona pellucida, and then finally it has to deliver this particular nucleus, leaving the tail out. So in a spermatozoa, what it matters is a nucleus of it. So often we say that spermatozoa is a DNA delivery boy. It does nothing except delivering a haploid set of genome to the next generation. But then along with this spermatozoa, then the theory suddenly changes with the evolution of the RNA biology. When we changed our focus from DNA to RNA, we say that spermatozoa does not only deliver you DNA, but it in fact delivers a whole host of RNA activities. In fact, the count says that there are about 18,000 different RNA molecules are delivered by the spermatozoa to the egg cell. And then these are all the functions. I mean, if you start reading, I'm not reading it for you. That includes everything that a new zygote need to do to be able to develop as a fully formed offspring. Along with the RNA and the DNA, the spermatozoa, it delivers a lot of proteins also. Out of that, the most important one that is worth discussing is the histone. And with the histone has to change to the protamine while the spermatogenesis process. And often this changing of the histone to the protamine, it creates a problem because it is not a 100% foolproof process. So whenever it is an incomplete change from histone to protamine, then the problem comes and the male is not able to do what it wants to do. Now we asked a question. I think this was sometime, it will come later. But can we identify a high fertile and the low fertile spermatozoa? What we did, we chose purposefully a particular locus, which is called H19 and IGF1. This is unisexually expressed. And then we collected the spermatozoa and took it through a bisulfite sequencing process to know their epigenetic status. And then what we do, okay, if you look at it, they are located like this, and then they have a DMR. DMR means the differentially methylated regions. And then these DMRs are again fortified by some sites which are called the CTCF binding sites. Anyway, let us come to the conclusion. When we look at this, we find that if we take a high fertile and a subfertile, we say HF and the SF, we find they remain differentially methylated with respect to their genes. And finally, let us not enter into too detail of this, but this is, we started. Okay, and then we asked a question. We asked a question that apart from a single allele, let us look at the whole kind of overall genome level methylation. We find that this thing happens. We had to design a particular chip for this, and then this is what we find. We find that the high fertile and the low fertile spermatozoa, the top three panels they remain the high, and the below three panels remains the subfertile. They remain distinctly differentially methylated for a specific region. Then we looked into which are these genes which are responsible for this, so that this becomes the molecule for the future. Then coming to, are some particular sperm smarter, because we know that about 1 million spermatozoa is deposited in the vagina. Ultimately, to the site of fertilization, it is hardly about 100 to 1,000 spermatozoa reaches over there. So what happens? How they reach? And then the small spermatozoa, we observe the microRNA involvement in this particular process. And then we started asking a question that at their site of fertilization, this is a male reproductive tract. So how these spermatozoa are coated by different protein? So based on the lateral evidences and the knockout evidences, we made beta differentiation as a particular molecule, and this is the whole protein map of the high fertile and the low fertile spermatozoa. We again find that they are differentially methylated. We located this particular proteins in specific regions, and then we looked into what is there in the sperm head. The sperm head is known to be very sweet. It has, this is why it's called the glycocalyx. That means a whole host of the forest of glucose, or the glycans, they are located over there. So we forget about this. We asked the beta differentiation, and then this is a set of experiment we did, where we are purposefully trying to take out the protein ports from the spermatozoa, and this is the lateral flow or the kind of the flow cytometer evidences we generated out of this. And finally, we propose this particular hypothesis. This hypothesis says that they are the pattern recognition receptor. These spermatozoa, which are very smart, they are completely armored with different glycans, so that they present before the neutrophils that don't kill me, I am self. Whereas the spermatozoa which are not, then they are being engulfed in the sperm, in the body of the uterus. So the question is, if this knowledge is to be transformed to a technology, we have to find a method by which we make we add these particular glycans over this kind of spermatozoa to make as a bad as a good. And these are the evidences where we see that if the spermatozoa, they remain incompletely encircled with the glycans, the macrophages, they attack them and then they try to kill them. You see here, then these glycans are of two kinds, the O-linked and the N-linked glycans. This is the high fertile. High fertile bulls, their phagocytosis rate is very less, whereas they are low fertile, their phagocytosis is very low, very, very high. So when you take out these glycans, particularly the O-linked and the N-linked, they start behaving like a low fertile bull. So this was a great lesson, what we understand, and then this becomes our target now, that how to change this low fertile semen to a high fertile situation. The molecular cargo of the seminal is, all the time we talk about the sperm, but we don't talk about the seminal plasma that contains, that makes about 90% of the semen is made by the seminal plasma. Am I correct? Yeah. So I think we need to relook at what is there in the seminal plasma. Incidentally, the seminal plasma, this carries a lot of EVs, or the extracellular vesicles, and this extracellular vesicle, as a truck, it carries along with this all the goodies that it wants to carry. It carries the proteins, the macroRNAs, and all that. And because they carry, that gives you a possibility that we can manipulate these EVs again, then to make them to uptake the spermatozoa if we want to transform this to a technology. And this is again a metabolomics kind of approaches which my collaborator, Dr. Kumarasan, he works at the root, and then finally they have to win over the cumulate, because when the spermatozoa reaches the oocyte, they have to penetrate to the cumulus cells. So then again, this is a receptor mediated phenomena, and we find specific evidences that 15 minutes. 15 over. Okay, I'll take 2 minutes, sir. So this is this NYBS8. This is another molecule which is found to be very effective. Then it has to crack open the zona pellucida, because they have to be they get friendly with the zona proteins that are present over there, and they are again a gene-mediated phenomena. So finally, probably this is what we expect from a sperm, and we said this is a dream sperm. What is this? They have to be good shape, good motility, good genetics, good epigenetics, good count. They are equipped to hide from where? Hide from the uterine tract, because we know the bulk of the spermatozoa there, they get lost in the uterine tract. And finally, they have to be equipped to win over the cumulus cells and the zona pellucida. In this lecture, I wanted to make these two gentlemen very famous. One is Pradeep, he is present over there, and her name is Renu. They are very prolific workers in our semen lab. But what is more important is this particular equipment, which is called a flow cytometer. Very soon, along with a kind of, you go to any semen lab, what do you find? You find a warm stage, you find a kind of the spectrophotometer, and you find the inverted microscope. So, but that does not give you a full idea about what you are freezing. But this particular equipment, I think very soon it will be a part of any semen lab. Now you need to qualify a particular semen. So this is evidence. Evidence is that a bull, which we consider as a good bull, but you look at the ejaculate to ejaculate reach. This is ejaculate 1, 2, 3, 4. They are mapped for the high membrane, mitochondrial membrane protein. And if you look at this red population over there, you see how the red population varies. For ejaculate 1, maybe in the month of January, this is 91.62, 45, 75, and 60. But had we had this information from before, we could have taken appropriate steps to ejaculate, to freeze it. So this is the take-home message. I said there is nothing like a good bull. So we should concentrate more on the ejaculate, differentiating the sperms for their content and the composition, not about only on their motility. More robust data we need to generate about how the season, the feed, the management, they affect this content of the sperm. So the possible intervention is, yes, we can directly approach the sperm We can also manipulate the seminal plasma. We can decide the site of insemination, and we can manipulate the female reproductive tract before we inseminate. So there will be the technology for the future for making possible to inseminate with a lesser number of semen, adding artificial competence to the poor sperm. I say if the sperm is poor, can we add competence to this and make them good? And then there may not be a single yardstick for the all bulls. When we freeze a semen, we take it granted that a single protocol will work on all bulls. It may not be true. We may have to generate a bull-to-bull different protocols. And finally, it looks like that it looks feasible to target 10 million spermatozoa per dose. If we do that, then without doing anything, we increase the semen production in this country by double, right? At two times. You understand that right now we have a shortage of the quality bull semen. If we can do that, then this straightway translates into improving the number of semen doses. Semen doses will remain. We will produce. But the question is from where we are making it, because the availability of the very high genetic merit bull is limited. All bulls, they are not high genetic. To fulfill the need and with a modest kind of ambition of inseminating about 35% of the animals that we are currently doing today, but in the Rashtriya Gokul Mission and the nation's ambition is to take it to 60%. So our demand for the frozen semen will increase. So these kind of thought process of the technology can make it better. So I stop here and I acknowledge this the fund of the ICAR and the DBT, Rabasam was kind enough, and the Bill and Melinda Gates Foundation for generating some of this data that we could make. Hope you liked it, and this is the kind of, I say it's a food for thought. So please think about this. I have not come with any prescription. I cannot give you a written prescription, you do this, don't do this. But I think you're all young researchers. If we join our hand together, I'm sure we'll do better. Thank you very much.
Thank you, Dr. for making a nice presentation.